again. Let me share my screen. Alright, now you should be seeing my screen. Let me remind you should check out the Canvas website and take a look at my site. I've got a link to my YouTube site, and I believe yesterday's lesson is up there. A link to the syllabus, which I'll show you in a minute. Some information about Zoom and some information about the lockdown browser requirement. Don't forget, you need to download the lockdown browser in order to, that didn't work. Well, let's click here. That's why it's not working. Some of these sites will not run inside of Canvas, and they'll give you a link, like right here, and you click on it, it then goes to my YouTube site in another window that's not within Canvas. Alright, so yesterday, or not yesterday, 2 days ago we're 7-6. So there's the lecture meeting. I don't see the lab meeting here. I'll look into that. Oh, here's the lecture meeting, and that's the lab and lecture meeting. I'm just reading down there. I know what that is. Okay. So everything is there. There's the link for the Avoiding Plagiarism tutorial. Remember, you must take that tutorial by Saturday and then take the Plagy quiz. You must get 100% on this Plagy quiz. You may take it more than once. I think it's actually something like 5 to 7 times. And although it's only worth 20 points, if you don't get 100 points on the Plagy quiz, you will get 0 on the infectious disease project, and that is worth 100 points. A guide for APA citations, and then a link to the study guide. You should take a look at that, because when you're studying for the quiz, you want to look at the objectives, and at the top of the study guide there will be objectives to chapter 1. Let me see if I can move that over. And then go through the objectives, and if you can answer the objectives, then you're doing well for the class. Why isn't that scrolling? Oh, there it is. There's a third one. And then in chapter 1, we also have chapter 10, and these are the objectives for chapter 10. And then after the objectives, I have the notes, which would be similar to the lesson, except these are sentences, and there are no pictures. It doesn't follow exactly the content of the lesson, but it has all of the information in the lesson. Actually, it is following a little bit how I went through it in the lesson. All right. Anything else I want to point out? Don't forget that the extra credit assignments, the study sheet is due July 10th, and the extra credit picture is due July 17th. I think that's this Saturday. And this is next Saturday. I mean, a week from Saturday. All right, we're not there, so let's go to week 1 and go through what we're doing. We're going to cover and hopefully finish chapter 1 and chapter 10 today. And then at the lab, we'll be doing lab 1, and the lab will begin at 6:30. Remember, I mentioned last time that we will spend 15 to a half an hour on lecture, and that's because when they made the summer schedule, they put more time in the lab, but they didn't put any more time in the lecture. And actually I need the time in the lecture, not necessarily in the lab. And you need more time because we only have 8 weeks in the summer, and we normally have 10 weeks. Let me see if that 8 weeks counts finals week. So it might be actually only 7.5 weeks. Oops, I lost it. Where is that? Yeah, 8 weeks counts finals week. So we'll only have 7.5 weeks. All right, any questions about what we're doing? If not, I guess I can leave that up for the lab. But let's go ahead and get started on the lesson. Oops, wrong one. Why didn't that work? For some reason, my program isn't working. Oh, there it is. I just can't see it. Took a while, or was it there and I just never saw it? So we talked about where is everywhere and where you can find microorganisms, and that's essentially almost everywhere on the planet. The exception is when there is lava or a volcanic vent. You won't have cells because the temperature will kill them. But even under freezing temperature, you won't have living cells, but they will be frozen. And once it thaws out, you can have life. Microbes are numerically the most abundant life form on the planet. How many microorganisms are there? And I'm talking about prokaryotic cells only. In 1 gram of soil, you can find 1 times 10 to the 8th cells. And this number will depend on how much organic matter is in the soil and how much moisture is in the soil. So this would be your average temperature and moisture, something like in western Washington or western Oregon. Not immediately after a rain, but there is moisture in the soil. If you had a very wet place with a lot of organic matter, it would be more than that. And if you had a very dry place like a desert, obviously there would be less bacteria. Do you find more or less bacteria in a milliliter of ocean water? And that would be 1 gram of ocean water. So more or less. Anyone want to guess? Come on, somebody guess.
Less.
Yes, you're correct. The deserts are known, the oceans are known as the desert of the planet. There would be less cells in ocean water than in your average soil. How about 1 gram of human feces? More or less?
More.
Yeah, you'd have more. Obviously, it depends on the condition of the human feces. If it's fresh out of the body, it would be more. But if you defecate in the desert and it's dried up for several days, it actually may be less, but that's because the bacteria have started dying from the desiccation. On 1 human being, you'd have more, and of course on the entire Earth, you'd have more than in 1 gram of soil. Let's take a look at how many you actually find. Like I said, a human feces, we say on average has 10 to the 8th, excuse me, that's not what I wanted. A soil, 1 times 10 to the 8th. Human feces, if it's very dry, you have 1 times 10 to the 6th cells. But if it's very fresh, directly out of the patient, you actually have 1 times 10 to the 11th cells per gram of human feces. And that is an awful lot of cells if you don't know. And actually, human feces, about 55% of the wet weight is from the weight of microbes. So the majority of human feces is actually microbes, assuming we're looking at wet weight. In a milliliter of ocean water, we have 1 times 10 to the 6 cells. And this won't vary very much, although you will have more cells if it's a very nutritious location, like where there's upwelling and maybe near the surface where there's lots of sunlight and oxygen. But it doesn't vary a lot. Maybe you'd have less in very deep ocean, as well as someplace where the nutrients are used up. A human has 3.8 times 10 to the 13th microbes, and that would be in or on the human, like in or gastrointestinal tract, on the skin, and that's an astounding number. And then on the entire Earth, it is 1 nonillion, which is 1 followed by 30 zeros. The human population is between 7 and 8 billion people, and so there's no question that there are a lot more microbes than there are humans. Any question about any of that? The point is just that there are astounding numbers of microorganisms. Now you should know that some microorganisms cause disease. And if a microbe can cause disease, we give it the term pathogenic. So a pathogenic microbe is one that causes disease. Most microorganisms do not cause disease. Only a small percentage of the microorganisms are pathogenic. Some of them are even beneficial to us, and we'll talk more about that later in the term, not today. When we talk about diseases, we should separate diseases into 2 categories. How are you guys hearing me today? I should always ask that. I noticed I didn't have that by my mouth, so.
I can hear you loud and clear.
Okay, good. So we should separate diseases into 2 types: infectious diseases and inherited diseases. Infectious diseases are caused by a microorganism. And can anyone give me an example of an infectious disease? Nobody's going to give me an example? Well, we'll move on. An inherited disease is one caused by somebody's genes. So when you're born, you may get an inherited disease. And what's an example of an inherited disease?
Cystic fibrosis.
Yeah, cystic fibrosis is dependent on the genes that you're born with. And if you're a cystic fibrosis patient, that means you have a gene, or actually 2 of them, for cystic fibrosis. An infectious disease is one caused by a microorganism, and some examples of that would be colds and flus, COVID-19, streptococcus pneumonia, or Streptococcus pyogenes causing strep throat. Okay? So those are just some examples. I won't test you on the examples, but you should know the difference between an infectious disease and an inherited disease. An infectious disease can spread from one patient to another. An inherited disease cannot, unless it's inherited, meaning a parent can spread it to the offspring, but you can't pass it to another patient, an inherited disease. Any question about that? We'll talk a little bit more about infectious diseases and if they spread. They're communicable if a disease spreads from one patient to another patient. Not all infectious diseases are communicable. We do consider Clostridium botulinum an infectious disease because it's caused by a microorganism, and in reality the microorganism is producing toxins, and then the toxin causes the disease. But my point is, it is caused by a microorganism, so we do call it an infectious disease, but it's very unlikely to spread from one person to another person. And the only example I can think of it spreading from one patient to another is with cannibalism, which, by the way, is a reason why you should not engage in cannibalism. Any question about any of that? All right, we're looking at different microorganisms, and you know they're small. How small is the average bacteria? Nobody's going to guess, so we'll say that they're small and they come in various shapes and sizes, and here we're seeing microorganisms of various shapes and sizes. To help you with the question, how small is the average bacteria? This is a bacteria, and there's a scale right there. Okay? So how small is the average bacteria?
I
50
can't read the.
micrometers.
Yeah, around 1 micrometer, which is a micron. Meaning those two terms are used interchangeably. That's the size of your average bacteria. E. coli, which looks this way, is actually a little larger than your average bacteria. And the width of it might even be more narrow than your average bacteria, but the length of it is, E. coli is definitely a little longer than your average bacteria. This is a fungus. That looks like a white blood cell, and that's forming a pseudopod right there. This is Volvox, which is actually a colonial microorganism. It is an algae, so it is a eukaryote, and it is green. And actually these little things here, that's a baby colony inside the parent colony, which is this thing here, which is sort of green. And you see each of these dots? That's a cell. Okay? And what will happen is this parent colony will open up, and then 6 baby colonies will come out of it, and then they'll go elsewhere and colonize the region, and they will eventually grow to the size of their parent, and then they will start reproducing. And then there's a white blood cell, which is microscopic. That's part of an organism, a human organism, so it's not really a microbe. But it is microscopic. That's about right. Microorganisms play a number of beneficial roles in the environment and for us. We use them to decompose organic waste in our water treatment facilities, and obviously they decompose organic waste all over the planet. Like when a tree dies, the minerals and nutrients in the tree are decomposed largely by microorganisms, which then make those nutrients available to other organisms. And when I said largely, you can also have mushrooms decomposing a tree, and a mushroom is not a microorganism. Microorganisms are producers in the ecosystem. Recall from your earlier class that a producer is one that makes its own food from carbon dioxide, and that can be in photosynthesis, turning light energy into chemical energy of sugar, or from chemosynthesis, taking the energy in a chemical and then using that energy to build up sugar. So converting it from one chemical to another chemical, which is oftentimes sugar. We'll talk briefly about chemosynthesis because bacteria and archaea are the only chemosynthetic organisms on the planet. Another beneficial role of microorganisms, we mentioned that they are involved in the cycling of nutrients and the nitrogen cycle, but humans use microorganisms to produce industrial chemicals like ethanol and acetone. When the farmer takes his corn and takes it make it into ethanol, the people who make ethanol are using microorganisms to make the ethanol. And I don't think there's any other way we can make ethanol. That's true for some other chemicals. You don't need to know this one, but we use microorganisms to make acetone. We use microorganisms to make some insecticides, and an advantage of these insecticides is that they are less toxic to humans as well as other animals. We use microorganisms to make fermented food. We've talked a little bit about beer and wine, but that's also true of leavened bread, meaning bread that has yeast in it, and that could be sourdough bread, which has yeast and bacteria in it. We use microorganisms to make cheese, to make vinegar, to make chocolate, and to make many other products. Okay, so all of our fermented foods use microorganisms to make that food. Microorganisms are used to make products used in manufacturing, like the enzyme cellulase is made by a microorganism, and then at a paper mill or some other company working with wood or paper, they can use this enzyme to degrade the wood. We use microorganisms to make antibiotics. For example, penicillin G, the first antibiotic ever discovered, is made by antibiotics. Some of the later antibiotics are man-made, like carboxycillin, which is a member of the penicillin family. It is not made by a microorganism. Somebody makes it in the lab. But many antibiotics are either made in the lab, like penicillin G, by a microbe, or they're made by a microbe and then somebody in the lab chemically alters the antibiotic. And that's something that we do with ampicillin. It's made by a microorganism, and then we take that and then we chemically modify it to make it ampicillin. Many other products are used by microorganisms, including some products in medicine. For example, human insulin does not come from a human. I mean, the gene initially did, but we don't grow it in humans. And when they make the insulin, they're making it in a microorganism. All right, any question about any of that? You should know that biotechnology is the use of microbes and organisms to produce foods and products for human use. Biotechnology is actually centuries old. And I don't know what the first event was, but it probably was the manufacturing of wine. where someone discovered that if you leave the wine out, microbes get in it, and then, not the wine out, the grape juice out, microbes get in it, and then ferment it to wine. Like I said, biotechnology is actually centuries old. We tend to think of it as something new, and there is a new aspect of biotechnology, that is genetic engineering, and it's simply a new technique of biotechnology. And this is new. Okay? Genetic engineering can be used to produce a variety of proteins from bacteria and fungi. And as an example, we use bacteria to make vaccines and other enzymes and human insulin. We use bacteria to make human insulin. We put the human gene that makes insulin from some human, we put that DNA into a bacteria, and then the DNA make the insulin. And then that's collected and purified, and then it's shipped away in a bottle, and then the diabetics use it. We use genetic engineering to engage in gene therapy. Gene therapy is when a patient is born, they have a defective gene, and gene therapy goes in and puts in the correct gene. Usually how that's done is you take cells out of the patient who has the defective gene, and then in vitro in a lab, somebody adds the correct gene to that cell, and then those cells are re-added to the patient. Anyways, that's gene therapy, to replace a missing or defective gene. And then we use genetic engineering to genetically modify bacteria. So we can modify bacteria to protect crops from insects or freezing by adding a specific gene. And let me see if I can think of an example with that. I'm afraid I cannot. I just don't know one off the top of my head where we added a gene and it protects the crop from birds and animals. One genetically modified tomato was the Flavor Savor tomato, and it was one of the first genetically modified vegetables or food, and it was a tomato that had a longer shelf life in the store, and so it did not rot, at least as quickly as other tomatoes did. And I think that's been a failure, because I haven't seen genetically modified tomatoes, meaning the Flavor Savor tomato, in the stores for many years. So I'm going to guess it was not economically viable and not enough people were buying it, and so the stores pulled it off the shelves, and it's essentially not sold anymore. That's my guess. When we're talking about microbes and their effect on human welfare, you should understand that they are important, and microbes create an ecology, meaning microbes are important on the planet for recycling nutrients on the planet, and this is part of the microbial ecology. And then microbes are also important with us. Like microbes help us digest our food. And these are the microbes living in our gut. So we talked about microbes and how they help run the nitrogen cycle, where nitrogen is recycled on the planet. But the microbes also help recycle carbon in the carbon cycle, and sulfur in the sulfur cycle. It is true that microorganisms do not have the same role in the carbon and sulfur cycle, as they do in the nitrogen cycle, where almost every step of the nitrogen cycle is happening because of the microorganism. In the carbon and sulfur cycle, there are many steps of those cycles that do not use microorganisms. Anyways, these are just some of the examples of microbial-mediated biogeochemical cycles that help support life on Earth, and without them, the nutrients would not recycle, and then they would just accumulate in the soil or in the ground or something, and they would not be available for other organisms. The first people to discover the microbial ecology was Winogradsky and Bajorink, and they were the first to show that microorganisms can recycle the nutrients of bacteria in the ecosystem. And we'll talk a little bit more about them. The reason why they were able to do this is because they did not study pure cultures. They would study bacteria growing in their natural conditions in the environment. And you need a microbial ecosystem before you can have the, or understand the carbon cycle, the nitrogen cycle, and the sulfur cycle. All right, any question about any of that? So the name you should know is Winogradsky, and he's the first to show that microbes were important in recycling elements and nutrients on the planet Earth. We use microbes to help us in bioremediation. Bioremediation literally means life, and to remedy, where we can use living organisms to deal with an environmental problem. And we've already discussed the environmental problem of sewage, but it also microbes are used with environmental pollutants. Like with an oil spill, you can spray microorganisms on the oil and have the microorganisms degrade the oil into carbon dioxide and water. So that totally will remove the oil and make it, I don't know, what's the word, non-toxic, so that other living things can use those nutrients. We can also treat other pollutant spills with microorganisms, and they will make the spill less toxic, such as a elemental mercury spill. If we treat it with the bacteria, it will convert mercury from elemental to some other form which is less toxic and harmful to humans and other microorganisms. I mean other animals. All right, any question about any of that? Well, let's go on to ways that humans can use microorganisms. We can use microbes as a biological insecticide. Sometimes they make the insecticide. Other times, like the example I'll give, the bacteria itself is an insecticide. And these alternatives to chemical pesticides tend to be less toxic than man-made chemical pesticides. But biological insecticides are used to prevent insect damage as well as disease transmission to crops. The example of a biological insecticide I'm going to talk about is Bacillus thuringiensis, bacteria where the infection is fatal in many insects. And here we're looking at Bacillus thuringiensis. And Bacillus thuringiensis is harmless to humans and other animals. It's also harmless to the plants. And in the 1960s, I remember my father actually bought Bacillus thuringiensis and then put it on the apple trees to prevent them from getting worms. So this would be a much better insecticide to apply to the apples, because you're not getting the chemical, I don't know, pollutant or the chemical residue of the man-made chemical that you're adding to the apples. Bacillus thuringiensis is an infection that's fatal to many insects, and of course, like I said, it's non-harmful to humans. What I wanted to mention was my father was a little lazy, and he did apply Bacillus thuringiensis maybe 1 or 2 times, and it did control the apples, the worms in the apples, for about 2 weeks. But like the man-made pesticide, you need more than 1 application of the apple to prevent the fly, it's actually a mayfly, you don't need to know that, from laying its eggs in the apple and then the egg hatching and then making the worm in the apple. Have I made myself clear here about Bacillus thuringiensis? I seem to have lost my train of thought, but the point is you need to apply it about every 2 weeks during the apple season, which lasts, I don't know, a couple of months. So you should apply it shortly after the apple tree blooms, and then you should apply it about every 2 weeks to make sure the apple never has a chance to get an egg laid on it to make the apple wormy. All right, any question about anything we've talked about so far? If not, we're going to switch gears a little bit and talk about the naming and classifying of microorganisms. This was started by this person here, who was known as Carolus Linnaeus. And he liked Latin so much that he Latinized his own name. I think his name originally in some Scandinavian country, it was probably Sweden, was something like Carl Linné. And he Latinized it to Carolus Linnaeus. And sometimes, like your textbook calls it, Carl Linnaeus, meaning only the last word is Latinized. He established the scientific nomenclature where every organism has 2 names. Every species has a genus name and a specific epithet for the species. So for the species Homo sapien, we have 2 names, Homo and then sapien, with sapien being the species epithet, but the species name is Homo sapiens, and Homo being the genus. This nomenclature, which is still used today, by the way, so the scientific community really liked what Carolus Linnaeus was doing, is called the scientific nomenclature, or the binomial nomenclature. It's used in science, so that's why it's called scientific nomenclature. And then it has 2 names to each species, and that's why it's called binomial, meaning bi for 2 nomenclature names. All right, any question about any of this? Scientific names are always italicized or underlined. So like when I'm writing it on the board, I would underline it to imply that it is a scientific name. The genus name is always capitalized, like on Homo sapien, The H is capitalized. And the specific epithet name is always lowercase. Like sapien, oops, didn't go there. Sapien, where am I? Sapien is always lowercase. Oh, there it is. I was on the wrong slide. That's the problem. The genus is capitalized. The specific epithet is lowercase. They're all Latinized, and it's used worldwide. So wherever you are on the planet, you can use the scientific nomenclature, and it would be understood by anyone who knows the scientific nomenclature. Even if you don't speak the same language, you could understand the scientific nomenclature because it's used worldwide. The name may be descriptive or honor a scientist, like Homo sapien is descriptive. It means man walking on two legs, and sapien means wise man. Okay? Escherichia coli is both honoring somebody as well as descriptive. Coli stands for, you can find it in the colon, so that's descriptive. Escherichia is named after Astor, which was an early microbiologist. Staphylococcus aureus is descriptive. Aureus means golden, and Staphylococcus aureus is golden or yellow. And then Staphylococcus means that the cells are shaped like a cluster of grapes, Staphylo, and then they're in balls, and that's what coccus means. All right, any question about any of that? After the first use of the scientific name, you may abbreviate the name by having the first letter of the genus and then a period, and then you follow with the specific epithet. For example, the first time you use Staphylococcus aureus, you have to spell it out, but all other times you can abbreviate it S period aureus. And then let me mention that Escherichia coli you should be doing that the same, but this one is so common that even scientists tend to call it E. coli. Because when we say E. coli, we know what we're talking about. Okay? And this one is so common that, well, frankly, it is used a lot more than Escherichia coli. So when we're naming and classifying things, you should understand that why we classify things is to group them in similar categories, meaning everything that's in the same group is similar. And before we talk a little bit more about classifying, we need to look at the classifying living organisms and how we classify all living organisms. Generally, the first classification system of all living organisms is the three-domain system. So we classify all living things into one of three domains. So all living organisms can be classified as a bacteria, an archaea, or a eukarya. Let's see if I can blow that up. Kind of. Nope, that's not going to work. Let's try it. Let's try it over here. There, that's better. I think I'm advanced. There we go. When we classify everything into the three domains, we sometimes call this the tree of life. The because the domains branch like a tree. And it is a multi-trunk tree, but it is a tree nonetheless. The classification of an organism into the domain depends upon the genetic makeup of that organism. So if we find a new organism and we don't know where to classify it, you just sequence its DNA, and then that will tell you where it belongs, because all of the bacteria are more similar to each other in the sequence of their DNA than another organism that does not belong in that domain. Did that make sense? So the domain is characterized for how we classify an organism, and it's based upon the organism's genetic data, its genetic makeup. There are 3 domains: bacteria, archaea, and eukarya, and they're all different from each other. In fact, it came as a surprise when somebody was sequencing the DNA of microorganisms that there was a domain archaea. We assumed incorrectly that these cells, which were prokaryotes, were similar to the bacteria. And actually they're very different. They're actually slightly, very slightly, closer to the eukarya than they are to the bacteria. Anyways, there's 3 domains. After the domain, you can separate the domain into kingdoms. We're going to use the 6-kingdom approach, where the domain bacteria is also the kingdom bacteria. The domain archaea is also the kingdom archaea. And then the domain eukarya has 4 kingdoms: Plantae for the plants, and Animaliae for the animals, Fungi for the fungus, and Kingdom Protista for the protists. Any question about that? Now, I should state that the definition of a kingdom, well, there isn't one. So there is no way to distinguish between what organism belongs in what kingdom. And there are debates about, for example, does an organism belong in the kingdom Protista, or does it belong in the kingdom Plantae? And the algae is actually one group of organisms that people argue about. Are they Protista or are they Plantae, plants? And we're not going to get into that argument, but you should know the kingdom is not defined, and there is no clear distinction between a kingdom, and then it's subjective. So some people argue about where an organism belongs. And I can say it's even worse than that, and that is scientists cannot agree how many kingdoms there are. We're going to use the 6-kingdom approach: kingdom bacteria, kingdom archaea, and these 2 kingdoms are the same as the domain. And then kingdom Plantae, kingdom Animalia, kingdom Fungi, and kingdom Protista. But there are other people who say there are different numbers of kingdoms. Some people say there's 5, some people say there's 6, which we will use. Some people say there's 9, some people say, I don't know, something like 15, some people say 20. When you get into the higher numbers, what usually we do, or what people do, is they start splitting this kingdom. Okay? And that's because this kingdom is not, it's not a real kingdom. What it is it is a eukarya that is not a plant, not an animal, and not a fungi. And if it's a eukaryote and it's not one of those three, we put it into the kingdom Protista. So the kingdom Protista is not sorry, I'm having a hard time saying this, not monophyletic. So that two members of the kingdom Protista may be different from each other, and that difference could be as different as the kingdom Protista to the kingdom Animalia. Any question about any of that? If not, let's move on. The bacteria have what the book calls a cell type prokaryotes. Archaea also has a cell type called prokaryotes. A eukarya have a cell type eukaryotes. And so the book calls these two different cell types. Some authors, obviously not Totora, will not call this a cell type. They will call it a super kingdom, meaning a kingdom above the other kingdoms. And you could call it a super domain because it's above the three domains. There's only two, what is that called? There's only two super domains. However, the classification of a prokaryote and the eukaryote has been around long before we ever knew about there being three domains. And that's why the cell type being a prokaryote is most often called a super kingdom. And that's because the kingdoms were around before the domains, and the prokaryotes and eukaryotes were also around and people knew about them, and they separated all living life things into a super kingdom. And I'm not going to talk about that further because your book does not discuss it. It refers to them as a cell type. Okay? But I'm just telling you that some authors call it a super kingdom, a classification of life above the kingdom. Any question about any of that? All right, let's talk a little bit about the domain bacteria. All the cells are of cell type prokaryotes. They have peptidoglycan in their cell walls. Most of the cells multiply by binary fission. And for energy, they can use any source that is available to living life forms. They can get their energy needs from an organic chemical, and we do that. We get our energy needs from the food we eat, such as glucose. But some of the bacteria can get their energy needs from an inorganic chemical, and that would be a chemosynthetic bacteria. And then lastly, some of the bacteria can get their energy needs through photosynthesis, converting the energy in sunlight into the energy in the cell, meaning the energy in glucose. Most of the domain bacteria are free-living decomposers or primary producers, but some of the domain bacteria are parasitic and some are pathogenic. Remember, a pathogen is a bacteria or a microorganism that causes disease. A parasite is one that lives off of the other and harms the other. So the other is harmed, it does get no benefit from the relationship, but the parasite gets a benefit. It gets a home and potentially nutrients from the person it's living off of. All right, any question about any of this? If not, let's move on to the archaea. They have the cell type prokaryotes, so they have prokaryotic cells. They do not generally have peptidoglycan in their cell wall. So the archaea do not have peptidoglycan. Some of the archaea are found in extreme environments, like you can find some of the archaea where there's methane, and we call those methanogens. Some of them live where there's extreme salt concentrations, and that we call halophile. Halo meaning salt, phile meaning lover, so lover of salt. And then there's the extreme thermophiles, and these are the organisms that live at elevated temperatures, much above our body temperature of 37 degrees Celsius. And these thermophiles live near the temperature of boiling water. Some of them can go above boiling water. Most of them are a little bit below boiling water. But all of them, well, not all of them, the range would be about 95 degrees Celsius for the average temperature they're living at. And although this is true, you can find the archaea in these extreme environments. When we look at every biome on the planet, we can find significant numbers of archaea. For example, they make up about 20% of the biomass in the Earth's soil, meaning of living organisms. They make up about 20% of the cellular weight. And that's true of plankton too, meaning the ocean water. About 20% of the weight of the microorganisms is from photosynthetic archaea. All right, the next question I have is, are any of the archaea pathogenic? Do the archaea cause human disease?
No?
Yes, that's the correct answer. Meaning correct, that's the correct answer. The archaea do not cause human disease. And for that reason, in this class, we will not be talking about the archaea very much. And generally, when I am talking about the archaea, I'll make it clear that I'm only talking about the archaea, and I'm not talking about the bacteria. And on this slide, we're only talking about the archaea. But none of them are known to cause human disease. And our focus in this class is on the microbes that cause human disease. And that's because most of you are going into the health care industry, and so we're going to try and focus on the microbes which cause disease. When we talk about the eukarya, I already mentioned that we can split them into 4 kingdoms: the plants, the animals, the fungi, and the protista. You should also know that all eukaryotes lack peptidoglycan. Of course, animals don't have a cell wall, and peptidoglycan is found in the cell wall of bacteria. But that's true of plants and the protists. They also don't have peptidoglycan. When we look at the fungi, I'm pretty sure I'm not going to ask you that you can find chitin in their cell wall. The fungi use an organic chemical for their energy needs, meaning they break down something and then use that organic chemical for its energy needs. And that could be glucose, but it could be some other nutrient, like when they're helping to break down a tree, that would largely be cellulose, and something would have to break down the something would have to break down the cellulose because the fungi cannot break it down directly. At least that's what I think. I'll have to look that up to be sure, but I believe you need bacteria. Yeah, I'm certain you need bacteria to break down cellulose, that only bacteria and archaea can directly break it down. But the fungi and the termites can live on wood because they're living in harmony with the bacteria, which is breaking down the nutrients like from the tree. The kingdom fungi always uses organic chemicals for its energy needs. The kingdom fungi can be multicellular, like the molds and the mushrooms, but they can be single-celled, and all of the yeasts are single-celled fungi. Some of the yeasts are decomposers, but some of them are pathogens, causing human disease. Any question about the kingdom fungi? If not, let's talk about the kingdom Protista. Remember, it's not a monophyletic group, and so we tend to break the kingdom Protista into three categories. The algae, which are plant-like protists, the protozoa, which are animal-like protists, and then there is a third category, the fungus-like protists. So the different protists can look like and act like a plant, and protozoa, an animal, eukaryote, or they can act like a fungus-like protist. Algae, of course, are photosynthetic, and they have cellulose in their cell walls. They produce molecular oxygen, and then they produce glucose, an organic compound, which is important for the planet because all animals get their glucose and their nutrients eventually from grass. Even if you're a beef eater and only eat beef, you don't eat plants, like you pull off your lettuce from the hamburger. Well, the cow got its nutrients from the grass, and the grass was engaging in photosynthesis. So all living forms on the planet ultimately need the photosynthetic organisms to make oxygen and various organic compounds like glucose. Any question about any of that? All right, let's move on to the protozoa, the animal-like Protista, and they are called animal-like because some of them can move. And these include the amoeba, the paramecium, and the euglena. All of these three can move. Let's skip this and talk a little bit more about moving. They move by one of three means. They can move by a pseudopod, shown here, and that's the way a white blood cell moves. They can move by cilia, which is actually, you can't see it, but that's how the Volvox moves. It has cilia, one for each cell, and then they all beat together, and that can move the Volvox colony. And then the third way they can move is by flagella, and that's a whip-like structure that beats, and then generally if you're beating this way, the object will move that way. So it acts sort of like a fishtail. Although instead of beating like a fishtail, it's more like snapping a whip. Some of the protozoa are parasites, like malaria, Entamoeba histolytica, which causes amoebic dysentery. That's throwing up and diarrhea, if you don't know. But not all of the protozoa are parasitic and causing disease in human. Some of them are free living. And I'm sure you've always seen like paramecium swimming around under your microscope. That would be a free living protozoa. And then, like I said, the third category of the protista are the fungus-like protists, and they will look like fungi even though they're a protista, and they will act like a fungus. All right, any question about the kingdom Protista? If not, let's talk about the kingdom Plantae. We're not really going to be talking much about plants because they do not cause human diseases, but you can separate the kingdom Plantae into 4 categories, into mosses, ferns, conifers, and flowering plants. And all of the members of the plants are multicellular. That is different from the protista, which are largely, let's go down here. I don't have it, so go over here. The protista are largely unicellular. All right, any question about the plants? If not, let's move on to the kingdom animals, the last group of the eukaryotes. We can classify animals if they're a sponge, if there is a worm, and this is a generic term. The correct scientific term is a helminth. Helminth means worm. We can separate the animals into insects, and then lastly we can separate the animals into vertebrates. We are a vertebrate, but so are dogs and cats and horses and a whole bunch of other animals on 2 or 4 feet. All of the animals are multicellular, so that differs from most of the bacteria, which are unicellular, and differs from many of the protists. Paramecium is a protist. Well, I'm not positive of that now that I think about it. No, I retract that. I can't for the life of me think of, off the top of my head, a protist which is more than unicellular, meaning that it contains at least 2 cells. I just can't think of any examples. So generally they're single cell. We will study some of the animals in microbiology. And what we're going to study in microbiology is a microorganism. And what is a microorganism? Well, all of the bacteria are microorganisms, all of the archaea, all of the vias and prions, and you should realize that these 2 are special because they're not organisms. They do not have a cell. Okay? But we still call them a microbe or a microorganism, and they can cause disease, but they're not a cell. And for viruses, you could argue about whether they're alive or not, and there are debates in the scientific community that we're going to ignore about that. But everyone agrees that a prion is not alive because all it is a protein molecule. And we'll talk more about prions near the end of the class. When we're talking about the protozoa, that's the microbe. Molds and yeast are microbes. Some algae, if they're unicellular, or if you need to see them under the microscope, we call them a microorganism. But some algae is very large. In fact, if you go along, walking along the Oregon or Washington coast, you'll find algae washes up. That is not a microorganism. So only some of the algae are microorganisms. And then there's one other category which we call a microorganism, and that is the helminths, the worms. Anyone have an objection with the worms? Are they microscopic? That's a yes or no. A parasitic worm, are they microscopic?
No.
Yeah, most of them are not. But microbiologists group the helminths into the microorganisms because the infective stage of all of the helminths, well, that's not true, of most of the helminths is the fertilized egg, which is microscopic. You need a microscope to see the fertilized egg. And most of the parasitic helminths, it's the fertilized egg which is infective to the human. The only exception, there are a few worms that can actually burrow into our skin, and those would be the only exceptions. And another reason why the microbiologists group the helminths in with microorganism is that the helminths can cause disease. If someone has a worm infection, that's going to cause disease. And the microbiologists study disease-causing organisms. And even though the adult helminths are not microscopic, I think the beef tapeworm is something like 6 meters long, definitely not microscopic. The microbiologists study it. Of the helminths, they're all multicellular animals. Many of them are parasitic, like the parasitic flatworms and the parasitic roundworms, but you should realize that most of the helminths are not parasitic. They will not infect a human, and they will not make a human sick. Any question about any of that? All right. So microbiologists study the helminths because the infective stage tends to be microscopic, and then the helminths also cause an infectious disease, meaning when one patient has a worm infection, they can spread it to other patients. So it is a communicable disease. And microbiologists always study infectious diseases. All right, we talked briefly about viruses. Oops, that's not right. That didn't work right. It should have gone only one time. I wanted to make it there, so actually that's pretty good right there, 90%. Viruses are special because this is a microbe that is lacking a cell, and so we call it acellular. It's also unusual because it does contain a genome, but that genome can be either DNA or RNA. It's never the two together. It's one or the other. And you should know that all other living life forms only have DNA as their genome. Viruses have their genome as the core molecule, and it's surrounded by a protein, and we call that the protein coat. And all viruses have the genome, DNA or RNA, and a protein coat. Some viruses may have the coat enclosed in a lipid envelope, but not all viruses have a lipid envelope. And when does a virus replicate? It only replicates, for that matter, it's really only metabolically active when the virus is inside of its host cell. So when it's outside the host cell, it is not metabolically active, it is not actively replicating. And it's only when it's inside its host cell that it can be metabolically active and actively replicating. You should also understand that viruses are not a part of the three-domain system. Let me go back to that. So if you look at this, you will see that there's no viruses in here. And that's because this is the classification of a living organism that has a cell. And viruses do not have cells, and the other reason is that they have very little DNA, and so it would be very hard to classify them based on their DNA. And that is true with viruses. We don't know exactly where they came from and how they're related to each other, and it's partly because they have such a small genome. All right, any question about any of that? So this is comparing the three domains of cellular organisms, the bacteria, the archaea, and the eukaryotes. The bacteria and the archaea have a cell type of prokaryotes. The eukaryotes have a eukaryotic cell type. The bacteria generally have one circular chromosome. That chromosome has no histone proteins on it, and there are no introns in the gene. And I'm sure you don't know, well, many of you do not know those terms. We'll be talking about them in the class. But what I mean by the bacteria chromosome is circular, when you begin it, I'll do it with this, so when you begin it right here, the chromosome ends at the beginning point, and so that's a circle. And so the bacteria chromosome are circular. That differs from a eukaryote that has many linear chromosomes. For example, on the X chromosome, right here, we have a starting plot and we have an ending plot. And those 2 points are in different locations, and that we call a linear chromosome. Now, I mentioned the bacteria do not have histone proteins on their chromosome. This is just a protein that you can find in eukaryote DNA, and it's also found in archaea. And when we talk about the chromosome, especially eukaryotic chromosomes, it has a very large proportion of that molecule, the chromosome, that is not DNA, that is protein. And most of that protein is histone protein. So histone is a protein. Now, bacteria do have other proteins on their chromosome, but they have no histone protein. I wonder if I want to talk about introns. An intron is a region of a gene that does not code for the gene product. For example, if you have the gene for lactase, the enzyme lactase, the gene encodes for the lactase enzyme. But there's a region of that gene that does not encode for the protein of the lactase enzyme. And that region we call an intron. And eukaryotes have in their genes regions of the genes that do not code for the product of the gene. And that region of the gene we call an intron. Bacteria do not have introns in their genes. Any question about any of that? All right, let's move on. The bacteria and the archaea have smaller ribosomes than the eukaryotes. The eukaryotes have an 80S ribosome. You don't really need to know this, but this is a size that the biochemists and the microbiologists came up with for ribosomes. And this is where the ribosome would sediment if you put them in an ultracentrifuge gradient and then put the gradient in the ultracentrifuge. So the eukaryote is a bigger ribosome and it sediments lower down in the gradient, and we call that the 80S sedimentation unit. Okay? The bacteria and the archaea have a smaller ribosome, and it sediments at the location in the ultracentrifuge gradient at the 70S location. And that S just means sedimentation. So this sediments at the 70S sedimentation area of the ultracentrifuge gradient. And you don't need to know any of that. All you need to know is that ribosomes of bacteria and archaea are 70S, smaller in size than the eukaryote ribosomes, which are 80S. And then I already mentioned about the cell wall. The major molecule in the bacteria cell wall is the molecules of peptidoglycan. In the archaea and the eukaryotes have no peptidoglycan in their cells. Any question about that? If not, let's move on. A brief history of microbiology. It largely, when we're talking about the history of microbiology, it largely pertains to the debate over whether or not microbes could spontaneously generate. And then the history of microbiology also refers to the golden age of microbiology, when microbiology was first getting started. And it's called the golden age because whenever a microbiologist studied something, it was something new. Nobody knew anything about it. And you do not need to know the years when I give a year, but the golden age of microbiology by our textbook author is considered from 1857 to 1914. And all you need to know is it's the start of microbiology. Now, this slide is showing you the major events in the history of microbiology. There were some events that had to happen before we could have microbiology start. And then when microbiology started, we had the golden age. And then there were, of course, important events happening after the golden age. Let me see if I can read any of that. There we go. In 1953, Watson and Crick published their model for the structure of DNA, meaning how DNA looked and a little bit how it functioned. All right. I have gone past time. I'm supposed to stop at around 6. I'm going to need about 20 minutes for this lesson to for the Zoom to finish processing the lesson. So I think I'll stop right here. And then at, let's see, 6:30, we're going to start the lab late today. Oops, I got the wrong slide. Because I need about 20 minutes for the, I don't know if I can write in this slide. Let's go to this one. I'm going to put start, well, I got to go to here. Just remember we need to discuss this slide. And it shouldn't be a problem because I'll be discussing it. And so let's meet back at 6:50 for the lab. And you guys, you got to remind me at 6 o'clock that we need to stop because the lab, I believe, starts at 6:30, but I need time for the Zoom file to process, because otherwise when I start the Zoom it can cause corruption of the first file and then it won't be available. So let's meet in the lab at 6:50. Any questions? All right, I'll see you at 6:50.